Welcome to the second session of my presentation. In this section, I will discuss about the heterocyclic compounds effective against. So in this, uh, as I told you that I'll discuss about the computational supported discovery of novel heterocyclic compounds effective against SARS-CoV-2. So if we go back 2 years ago, and we can imagine that the whole world was panicking because of the COVID-19. So coronavirus, if we see, it was evolved as first identified human coronavirus in 1965, and it caused a common cold. There are mainly 7 strains of coronavirus which are known. Some are mild and some are fatal. So the most fatal one we observed recently is SARS-CoV-2. So the genera which is present in coronavirus is beta coronavirus, 16 non-structural proteins and 4 structural proteins. The structural proteins are mainly spike protein, envelope protein, membrane protein, and nucleocapsid protein. And this coronavirus mainly target and it attacks the human cells at multiple points, mainly lungs and throat. It belongs to a family of Coronaviridae. Coronavirus, there are various targets already identified for coronavirus. And these targets are nothing but proteins which are responsible for the survival of the coronavirus. So if we look at the structure of this coronavirus, as I told you that it contains structural proteins and 16 non-structural proteins. So the drug targets in coronavirus could be a spike protein, proteases protein, polymerase or helicase protein, or capping proteins. If we look into the drug discovery process, we all know the drug discovery process is a lengthy process in which it requires drug R&D and clinical trials. So this is divided into 2 phases, and after clinical trial, it goes for the FDA review and approval. So the initial process of drug R&D is the selection of, or you can say the identification of the disease. When the disease is identified, then it go for target identification and validation, then hit generation, then lead optimization of that compound by various studies like potency study, PK, ADME profile study, SARP pharmaco modeling. And after this, the compound is synthesized and go for preclinical and animal studies. When the compound crosses this stage, this whole stage takes 5 to 6 years. And when this crosses this stage, it enters into the clinical trial where it faces 3 phases, and it is directly tried on people. And after studying its efficacy and safety, it is go for FDA review and approval. Unfortunately, at any stage of this, the compound could fail. And what happens, and sometimes it also happens that the drug which passed through all these preclinical and clinical trial, it fails at the final stage. If I give you one example, the example is remdesivir. We all know that during pandemic, drugs which was approved by FDA for emergency was, one of the drug was remdesivir. And this remdesivir, it is basically synthesized by Glide Sciences USA, and it is a broad spectrum antiviral which was synthesized to treat Ebola virus. And the mode of action which is there where it attack is RNA dependent RNA polymerase enzyme. Now drug, obviously this drug is effective, and if we see the efficacy profile of this drug, it is effective, though it is an effective drug, but highly toxic. And because of this toxicity to the hepatic activities, this was failed at the last stage of the drug discovery. And now FDA and WHO banned this drug for the use against COVID-19. So this is one of the example. The another example is hydrochloroquine. Hydrochloroquine, we all know, is basically used to treat malaria and arthritis. And in the year 2020, FDA issued concern because of the increased number of reports showing serious heart rhythm complication in patients treated for COVID-19. So this drug was also failed at the latest stage, and now WHO also banned the use of hydroxychloroquine. So there are some other examples like lopinavir and ritonavir, which was earlier the HIV drug, and now it basically causes dysfunctioning and adverse effect on liver and gastrointestinal problems. So that this is also banned. So in the year when it was pandemic and the COVID-19 swapping the world's economy, and people throughout the scientific community were looking for the therapeutics effective against COVID-19. So what happens was, in the absence of sufficient information about SARS coronavirus 2, the computational tool become rapid and effective pathways in this area. And there was a strategy, that is the repurposing of the existing drugs. So our approach toward the management of COVID-19 was also the same, that is drug repurposing approach. What we thought of doing at that time, when the data was insufficient, then that we took a library of compound, we did its in silico studies, and we tried to analyze that which existing drugs could be repurposed. So there was a paper in Scientific Reports by Cotard in 2020, and what they did is they took the library from Prestwick, and they took 15-20 approved drugs, and what they did is they did the in vitro screening in an infected cell bias assay, and out of these 15-20 approved drugs, 11 compounds showed EC50 value in the range of 2 to 20 micromolar. And out of these 11 compounds, the two compounds were the hit compounds, that was hesperidine and diosmin. And now these two drugs, they are in clinical trials. So these are the structure of hesperidine and diosmin. And in this case, we can see that there are so many hydroxyl group present. So the effectiveness of these drugs could be attributed to the hydrogen bonding between the protein and this ligand. Now, targeting multiple enzymes of SARS-CoV-2 is a potential approach to combat COVID-19. And we also followed this approach. We selected few targets like NSP3, NSP9, NSP12, and NSP15. Why? Because they share 83% similarity with the existing earlier reported SARS-CoV, and the data is available with us. So our approach was what we did is, after reading so much of literature, we choose these four proteins in, and then we take a library of 15-20 compounds, and it was observed, and we choose we did the virtual screening of all these 15-20 compounds against these targeted proteins. And it was observed that the best hit against each targeted protein was hesperidin against NSP3, diosmin against NSP9, catenuolin against NSP12, and acarbose against NSP15. We observed and identified them as the best because of their best docking scores and best binding free energy. Then we found other hits against these targeted proteins. So apart from hesperidine, the second hit was diosmin for NSP3, hesperidine for NSP9, diosmin for NSP12, and catenuolin, hesperidine, and diosmin for NSP15. So after these docking score and after these observation, what we analyzed is diosmin was the one compound which was showing good affinity with all the targeted proteins. And that means this diosmin is showing multi-targeting behavior, and it is showing good affinity with all with all the four proteins that we analyzed. So with NSP3, NSP9, NSP12, NSP15, the docking score and the binding free energy was found out, and it was observed at then we go for further analysis. Then we took this diosmin for MD simulation studies, and these MD simulation studies analyzed and revealed that better stability of ligand and protein complex was there with diosmin. So this work we published in Methods against an invitation from the journal in 2021. And now after taking the known library of 15-20 compound, after publishing this work in Methods, we analyzed that our approach should be to build an in-house library of 25 compounds. So what we thought of was, we take the most potent antiviral drugs, we explore them against MPRO of SARS-CoV-2, and we take their docking score. And according to the docking score of all the potent antiviral drugs, we found that indinavir was the best one. So the docking score of indinavir was come out to be the best one. So we took indinavir as the positive control amongst all these potent drugs. And if we see the structure of indinavir, so indinavir is already approved for the treatment of HIV. And if we see its structure, it contains this hydroxyethylamine moiety. So based on this moiety, we designed an in-house library of 25 compounds. And all these, 2,500 compounds. And all these 2,500 compounds were screened virtually. And out of all these 2,500 compounds, 25 compounds showed better results as compared to indinavir. So we take out all these 25 compounds, and these are the structure of 25 compounds. And out of all these 25 compounds, the compound 16 was found to be the hit compound. How we found out that compound 16 was the hit compound? By studying, by doing in silico studies, and by investigating the docking score and XPG score. So the docking score of 16 was the average one, the best docking score was found to be for the compound 1, that is minus 9.864. Then after investigating the docking score and XPG score, we did the ADME analysis of all these 25 compounds along with 26 and 27, that is indinavir and atazanavir. So after seeing the physicochemical properties of these HEA-based drugs, we see that it is only this compound number 16 which is showing no violation against the Lipinski rule. And if we see other compounds, there is either one violation or two violation. Even indinavir and atazanavir, they are also showing violation. So we choose this as the best compound with no violation of Lipinski rule. So we compared the docking scores, and when we compare the docking score of indinavir, which is minus 8.824, and compound 16, which is minus 8.955, this suggests that the compound 16 has an ability and it is adhered to drug-like parameters. Then what we observed, we did deep studies regarding that, and we compared the interactions of the protein and compound protein ligand interactions of indinavir and compound 16. And these were the amino acid residues which were interacting with indinavir either through hydrogen bond or salt-salt bridge or pi-pi interaction or hydrophobic residues. And these are the amino acid residues which are interacting with the compound 16. And the protein which we use for docking is CL-Pro. So it has been observed that indinavir do not show any pi-pi interaction, whereas compound 16 do not show any salt bridge residue interaction. So that indicates that the stability of the compound 16 in the pocket of protein CL-Pro was much better, more in comparison to indinavir. So after studying these 25 compounds, we also designed another 8 compounds to study the SAR analysis. And all these compounds are based on the structure of compound 16. These are modified compounds in which we introduced the CF3 moiety or fluorine moiety at different positions of this pocket 2 or pocket 1. Still after this analysis, we observed that compound 16 remained the best compound amongst the library of these compounds. So it has the best docking score of minus 8.955 in comparison to all other compounds. Now, after this, we again, from docking score to ADME profile, then we go to study the MD simulation studies. And the MD simulation studies again revealed that this compound 16 shows stability in the pocket of the CLO protein, and there is a stable ligand protein complex formation. And they are stable up to 100 nanosecond simulation time. So after designing this in-house library of compound, selecting the hit compound, now the next target is to synthesize such compound. And for the synthesis of such compound, what we did is we optimized another method. We took this as a commercially available compound. We did the ring opening reaction using different amines. And in general, in the literature, this reaction was carried out in presence of isopropanol for 12 hours. But we optimized this reaction in microwave, And what we observed that the product with up to 98% yield was observed within 20 minutes. We tried to change the solvent also, and we observed that the best yield was formed in nitromethane. So nitromethane, we all know, is quite toxic. So we replaced the nitromethane also by using a green solvent, that is ethanol. So we have seen and then we studied the reaction mechanism in detail and the effect of these solvents on this reaction. We studied in detail and we did lots of studies related to NMR titrations. So we observed that this reaction is a one-pot green synthesis. It plays a dual role of, so the nitromethane here plays a dual role of a solvent as well as catalyst. This is energy efficient reaction because we are doing it in microwave, a very easy workup and less time is consumed in this reaction. We synthesized library of compounds by varying different R groups here, and then we optimize this reaction using different solvents, using nitromethane, DMSO, ethanol water combination, DMF, isopropanol, and we also vary the molar ratio, 1:1 ratio and 1:2 and 2:1 ratio of the reactants. We also varied the power and time, and what we really observed was the best result was obtained when the 1:1 ratio of the compound were taken of epoxide and amine, and the reaction was giving 90% yield in nitromethane at 300 bar of microwave within 20 minutes. So if we compare this with ethanol, which is a green solvent, at the same condition, the ethanol was giving 70% yield. Then different effect of electron withdrawing group and electron donating group was also analyzed to compare the yield, and it was observed that electron donating group present on the amine was giving the better yield in comparison to electron donating group because of the ability of the amine. The lone pair present on the amine can easily attack when this lone pair is available, when electron donating group is there, so it can easily attack this epoxide and opens it up to form the product. So after this, the curiosity was in our mind that why nitromethane, why in presence of nitromethane, the yield was high. So we tried out to find out the problem and this reason by doing computational analysis, as well as we perform the 1H-NMR titration. So what we did that we initially take the NMR of 2A of 2A means this 2A is this 2A, that is aniline. So what we did that we take the NMR of aniline and then we take the second. So this is the NMR of aniline, and then we add nitromethane into the aniline. So when we add nitromethane into the aniline, that is the NMR B, this which is shown in red, we observed a shielding of aromatic protons and the methyl proton. So this shielding of aromatic proton and methyl proton shows that the nitromethane interacts first with the hydrogens of this aniline and causes this shift. So it basically catalyzes. It basically, after interacting with this, it makes the lone pair of electrons available on nitrogen so that it can easily attack to the epoxide ring and it can form the product. So this helps us to analyze these reactions, and these studies help us to analyze why nitromethane was giving the best results. So this was all about this synthesis of these analogs, and this was published in ACS Omega in the year 2020. Then again, we do not stop ourselves at the point of synthesis. Again, in the similar way, we were trying to build another in-house library, which was effective against NSP 15. So this NSP 15 is the Nendo U enzyme, and we have created a library of compound, and in that compound, compound 4 was found to be the identified hit. So after the virtual screening, this compound was found to be the hit one, and the control which we took in that case was tipiracil. So after our findings that this is a hit compound, we synthesized this compound by the method, by following the method which we published in ACS Omega, and we synthesized this hit compound. And after obtaining and purifying this hit compound, after characterization, we go for biological assays. So we have performed 2 biological assays in this: SARS-CoV-2 entry assay and SARS-CoV-2 spread assay. And during this biological assay, we took ivermectin as the control. Now, it has been observed that the ivermectin, we found that compound 4 was showing better efficacy in the entry assay. So if we see the IC50 value, it is 4.973 micromolar in entry assay, and it is 7.289 micromolar in the in case of ivermectin. So our compound was showing better efficacy in entry assay in comparison to the spread assay. So these were the results we obtained, and we published this in RSC Medicinal Chemistry. Then again, there were, we all know there are, after looking at the inhibitors, after having so much of literature survey, we designed another type of molecule in which we designed this compound 5. And why we designed this compound 5? Because we have analyzed that this compound was showing very good binding score of minus 45.86. But the problem with this compound was that it was not following Lipinski rule because of its size and higher molecular weight. So we tried to modify this compound 5, and what we did was, we modified this, and we break the C2 symmetry of this compound. And we, after this, we'll get 2 compounds, compound 7 and compound 8. And then we also try to do the in silico analysis of compound 7 and 8. The compound 7 was found to be minus, and the binding energy of compound 7 was found to be minus 55.72, and of compound 8 was found to be minus 52.14. So the values were quite higher than that of compound 5, but still, because these compounds were following Lipinski rule, we synthesized them. So we synthesized these compound 5, 6, and compound 5, 7, and 8. We purify them in our lab, and then we characterize them. And after characterization, we again go for biological assays. So the 3 compound 5, 7, and 8 were tested, and we did the spread assay, and the control which we took was ivermectin. So it has been observed that the IC50 values in micromolar range were 14.71 for compound 5, 10.05 for compound 7, and 12.46 for compound 8. So all these values were higher in comparison to ivermectin. That showed that the compound 7 was, amongst all these three compounds, 7 was the best one, but it was less potent than ivermectin. So these were the results of SARS-CoV-2 spread assay, in which we plotted this graph, and this paper was also published by our group in BMC 2021. So if I look into the overall strategy which we are following for the management of COVID-19. So what we were doing, but from where we have started with the structural based designing. Then we followed the synthetic approach. We optimized the synthetic strategy. We use microwave assisted synthesis of identified hit against SARS-CoV-2. We published this in ACS Omega, and we did green synthesis, which is energy efficient, microwave assisted synthesis. And now also we are trying to optimize certain reactions which could be mechanochemical. So one of our students, she is working on the mechanochemical series of reactions, which has easy workup and less time. The another thing which we did after synthetic approach, we found out different inhibitors of different proteases of CL-Pro and NANDO-U, and we found out that these were the best hits by virtual screening, and then we synthesized them, and we did the biological assays, and these were found to be found to have good biological results. Then now we are also working on a library of 20 compounds, which is synthesized, and this is the generic structure of the compound. And all these 20 compounds are non-toxic, and they are IC50 coming in the range of 2 to 5 micromolar. So with this work we are doing in collaboration with INMAS, and we are hoping that we will get good results soon. So this work is basically, I will give a full credit to Sumit, who is my PhD student, and he is a hardworking boy who is working on COVID-19, and who thinks and who discuss nicely with me, and he is a hardworking fellow, and I always wish best. I always give best wishes to him for his work. So these are important papers in high impact factor journals, which we published recently. Then, so I'll now come to the last stage of my my presentation. I would like to acknowledge Miranda House, which is giving me platform to work, to do my research work. And I would like to tell you that we all know that Miranda House is NIRF #1 from last 5 years. And from 2017 to 27, we are proving ourselves by doing hard work, and we wish that we will maintain this position always. I would like to acknowledge our collaborators, Dr. Brijesh Rathi from Hansraj College, Professor Agam Prasad Singh from NII Delhi, who is basically helping me to pursue the biological work. So Charu, she is working in his lab also, and she is doing biological work in his guidance. I would also like to acknowledge Aniko Borbas. She is my Hungarian collaborator. So we had one Indo-Hungarian project, and she is Professor Nidhi Jain from IIT Delhi. Apart from them, I'm also working with Dr. Prakasha Kampaya. He is currently in Mayo Clinic. Professor Miguel Prudencio from Portugal. So these are the people with whom I am working, and I'm extremely thankful to them because they are the people who are helping me uh, in the college at such platform where the facilities are less. So for it's really difficult for a medical chemist to survive in colleges because of the lack of facilities. So I am extremely thankful to all my collaborators. I'm also thankful to the funding agency. So in the last 6 years, we received a grant of more than 2.2 crores. And I'm thankful to UGC, DST, and SERB for giving me an opportunity to fund for my research work and helping me in this way. The last but not the least, my students, Charu, Sumit, Neha, Meenakshi, and they are all my undergraduate students, Rupal, Sheetal, and Palkaran. Now currently they are working in IITs and they are pursuing PhD. I'm so happy that a, a little encouragement is essential at undergraduate level to do good in their career. So they are doing really good. And if I talk about my PhD students, they are also working hard to get the best result. So this is my research lab in Miranda House, Chemistry Research Laboratory. So I'm extremely thankful to Professor Vijay Lakshmi Nanda, who is helping me and who is giving support to carry out my research work in Miranda House. So last, I would like to end here with this quote that coming together is a beginning, keeping together is a progress, and working together is a success. So I always believe in working together, and one more most important key is the collaboration. Collaboration is a key. It takes innovation and creativity to the next room. So I always believe that alone you can't do much, but when you do work in unity, together when you do work, and you open the space for success. So thank you so much for listening to me, and all the suggestions, all the questions are welcome. I would be happy to answer your queries. Thank you so much.